Every living being in this universe, right from a tiny microorganism to man, are subjected to multiple invasions all along by some other living organism or the other. Every organism establishes a sort of equilibrium with its environment. Our body and its outside environment and the organisms, we all form a sort of equilibrium with each other. But very often this equilibrium is upset and the invasion takes place. Fortunately, among the existing millions of microorganisms, we have only around 400 pathogenic microorganisms. Out of these again, we have only 150 microorganisms which we come into contact, and further only around 10 infections or so we pick up. Is it not fortunate? But then even these infections have to be fought. How do we fight these infections? Every individual, every living being has a defensive mechanism of its own. Every organism has to fight for its life. These defensive mechanisms have to be encouraged and kept ready. That's exactly what we do by vaccination. By vaccination, what we do is create a sort of artificial war atmosphere by introducing some dead organisms or those organisms which have been rendered useless are made to create an atmosphere inside the body simulating the aggression, and the body in its response produces certain defensive bodies which will fight in the event of actual invasion. That is exactly the subject of immunology. You must have heard the proverb, prevention is better than cure. Nothing could be more truer or more important than this axiom. This exactly forms the essence and subject matter of immunology and vaccinology. Different immunization procedures that increase the resistance to infectious agents justify Edsall's statement that never in the history of human progress has a better and cheaper method of preventing illness been developed than immunization at its best. Vaccine, which is being utilized to immunize the animals and birds against various diseases, forms the major part of immunology. Production of vaccine now described is categorically grouped into viral vaccines and bacterial vaccines. Among viral vaccines, a vaccine developed in tissue culture, a vaccine produced by chicken embryo inoculation, and a vaccine produced by intracerebral inoculation are dealt here. Let us first go through the tissue culture rinderpest vaccine. It is a viral vaccine produced against rinderpest disease. This rinderpest disease is characterized by shooting diarrhea, rise of temperature, and finally the death among cattle, sheep, goats and pigs, etc. The disease is something like AIDS in human beings. Here the virus destroys lymphoid tissue, which is the very source of cells involved in the immunity. Fortunately, we can produce a very potent vaccine against this disease. For production, calf kidneys are collected aseptically in a sterile beaker. Then in the laboratory, the capsule of the kidney is removed, followed by washing with a chilled phosphate buffer saline 3 times. With the help of scissors, the cortex portion of the kidney is clipped and placed into sterile beaker with phosphate buffer. Then it is finely minced and the material is transferred to the trypsinization flasks. About 50 ml of 0.25% trypsin is added and allowed to blend on magnetic stirrer for about 10 minutes. After digestion, the material is transferred to centrifuge tubes and spun at 600 to 700 rpm for 10 minutes. The supernatant is discarded carefully and chilled phosphate buffer is added to tubes containing these cells. The suspension once again is centrifuged for 10 minutes at 600 to 700 rpm. Again the supernatant is discarded and the growth medium is added and centrifuged for about 5 minutes. After discarding the supernatant, the packed cell volume is noted and the cells are diluted at the rate of 1 in 200 in growth medium. Now, the liquid seed material with a strength of a 5 log per ml is added to 1 liter of medium containing suspended cells with a concentration of 2 log per ml. It is mixed thoroughly and transferred to the Rook's flasks at the rate of 100 ml per flask. Finally, the flasks are shifted to incubator. These flasks are incubated for 24 hours and later checked for any signs of contamination. The field is screened for growth and division of cells. After 72 hours, we can find complete monolayer in the Rook's flask. After this period, we can find initiation of cytopathic effect, which becomes evident from the rounding of the cells and the development of refractivity. The medium is discarded after 3 days and 100 ml of freshly prepared growth medium is added to each of the controls and the infected flasks, and the flasks are once again incubated. Every day we have to examine each flask for cytopathic effect and any possible contamination. As cytopathic effect advances, we can notice swollen cells, elongation of cells, disorganization of monolayer, giant cell formation, syncytial formation, and finally detachment of monolayers from glass surface. It takes about a week for completion of cytopathic effect. When there is about 90% of cytopathic effect, we have to transfer the flasks to refrigeration. Today, this raw vaccine is collected and a stabilizer is added at equal quantities. Then penicillin and streptomycin are added and finally titration is carried out. We should get a titer of 5 log or more. After titration, the material is sent for freeze drying. Freeze drying at -40° centigrade should be completed within 3 to 4 days. After freeze drying, the random samples of a freeze dried vaccine are sent to the standardization division for conducting sterility, safety and potency tests. The vaccine is released for field use only after passing all clearance tests from the standardization division. Now, let us go to another vaccine, the Newcastle disease vaccine, which is also known as Ranicate disease vaccine. It is being produced on chicken embryo. This Ranicate disease is a common disease of poultry causing heavy mortality. The disease is comparable and related to influenza of human beings. It is a serious problem faced by poultry industry. Fortunately, we can produce a very effective vaccine against this disease. The seed virus used here is Comoros strain. Fertile eggs are obtained from salmonella free stock and set in an egg incubator as for hatching of chicks. The eggs are turned at least twice a day, and on the 7th day of incubation all the eggs are candled. Any infertile egg or those with dead germs are discarded. Again, on the 10th day of incubation all the eggs are candled and selected for inoculation. Only those eggs whose embryos are quite active with vigorous movements are picked up. At the same time, a mark is made on the shell at the selected spot for inoculation to avoid blood vessels. For sterilization, a swab of tincture iodine is applied to the marked site of inoculation and also at the central spot on the shell covering the air sac. A small cross of 3 millimeter dimension is made with a dental drill at the site of inoculation. A puncture at the center of the air sac is also made with a sharp needle on all the eggs. Now, the eggs are ready for inoculation with seed virus. These eggs are inoculated with 0.1 ml of 1 in 100 dilution of seed virus, that is Comoros strain. The inoculation is made into the allantoic cavity by piercing the shell membrane at the cross already drilled. The puncture made at the site of inoculation and the air sac are then sealed with paraffin and then again transferred to the incubator. The eggs are candled after 24 hours of incubation. The eggs with dead embryos are discarded. After 48 hours, the eggs are again candled and all the eggs with dead embryos are collected for harvesting. The eggs with dead embryos are chilled for 30 minutes in a refrigerator at 4 degrees centigrade. The chilled eggs are then taken for harvesting and kept in a hood which has already been sterilized for 1 hour with ultraviolet irradiation. The eggs are then opened at the air sac. Hemagglutination spot test is done with a small quantity of amniotic fluid collected. When the hemagglutination spot test is satisfactory, all the fluids and the embryos are collected from the eggs into a sterile beaker which is kept cool. To the pooled fluids and the embryos, necessary antibiotics, that is penicillin 1000 international units and streptomycin 2 to 3 milligrams per ml are added to prevent any accidental bacterial contamination. Then the whole material is blended in a sterilized mixer for 5 minutes, cooled and again blended for the same time. The blended vaccine material is then stored in a refrigerator. Now the material is ready for ampouling. The beaker containing the suspension is taken out of the refrigerator at the time of filling and sterilized ampoules are kept cool on ice. The sterilized ampoules are then filled with 0.5 ml quantities of suspension with a sterile syringe. The vaccine in the ampoule is now ready for primary freeze drying. The vaccine ampoules kept in a specially made container are then loaded in a primary freeze dryer. The primary freeze drying is done for 18 to 20 hours, during the course of which the water vapor escapes till the vaccine becomes a dry solid slant. Then the ampoules are removed from the primary dryer. After the completion of drying, these ampoules are sealed using hydrogen gas flame under vacuum. Then the ampoules arranged in the tray are stored in deep freeze at -20 degrees centigrade. The ampoules are sent for quality testing. Only after the vaccine passes the safety and the potency tests, it is released for field use. Now we will deal with a vaccine based on the intracerebral inoculation, that is Semple's anti-rabic vaccine. Rabies is an important viral disease of zoonotic importance which is causing concern to the veterinary and the medical profession. The vaccine produced at the Veterinary Biological Research Institute at present is the Semple's vaccine. For preparation of the vaccine, young healthy hornless sheep of 6 to 8 months age are selected. At one time, 30 to 40 sheep are inoculated intracerebrally with 0.5 ml of 1 in 100 to 1 in 200 dilution of seed virus, that is Pasteur's Paris strain of rabies fixed virus in a single injection. Sheep after inoculation show the symptoms like rise in body temperature, mydriasis, trembling and sluggishness, followed by definite signs of paralysis. Sheep showing above symptoms are taken and sacrificed. Then their heads are separated using hammer and chisel. The skull is broken and the brains are collected aseptically. The harvested brains are blended in a mixer and 1% phenol in distilled water is added to make 10% brain suspension. This is incubated for 48 hours at 37 degrees centigrade for inactivation of virus and agitated at 12 hourly intervals. After incubation period is over, the suspensions are diluted with equal amount of double strength phosphate buffer solution and kept for 14 days at 2 degrees to 5 degrees centigrade. Then it is filtered through several layers of muslin cloth for removing any particulate brain tissue. The same is subjected to various tests after dispensing in sterile bottles. The vaccine bottles are in presentations of 210 ml container and 35 ml container. The vaccine is released to field use only after clearance is given by the standardization division. Now we will discuss about bacterial vaccine. Hemorrhagic septicemia is a disease of cattle where it affects the respiratory tract and causes septicemia and finally death. It is comparable to human plague. It is a serious problem in cattle and buffaloes. The vaccine against hemorrhagic septicemia, which is caused by Pasteurella multocida, is produced at Veterinary Biological Research Institute, Hyderabad. The strain used for vaccine production is Pasteurella multocida P52. The seed culture is maintained on blood agar slants. The bacterial culture is inoculated into the nutrient broth and incubated for about 18 to 24 hours till a luxuriant growth of the organism is obtained. About 1 to 2 ml of this culture is again inoculated into 3 liter production flasks containing same nutrient broth and again incubated for about 18 to 24 hours. The broth with culture is again formalized to inactivate the organisms by adding about 0.5% of formaldehyde. This is once again incubated for further 24 hours for stabilization and complete inactivation. The formalized culture is pulled into a container with stirrer where 0.5% of potassium alum is added. Finally, the pH is adjusted to 7.2 with sodium hydroxide. Now the vaccine is ready for bottling. Here bottling is done with automatic bottling machines under strict aseptic conditions. The vaccine thus prepared is ready for field use after thoroughly passing through the various sterility, safety and potency tests at standardization division. Vaccine is a powerful tool in the hands of public health authorities, the medical authorities, the veterinarians. It is the vaccine with which we can combat any infectious disease. Have we not eradicated smallpox from the face of globe? It is because of the vaccine, the potent vaccine. How the vaccine protects the animal or the man? The vaccine exactly simulates the infection, but It contains only dead organisms or the inactivated organisms. It is something like dummy training, something like preparedness in the war. For war games, we have always such exercises with the dummies. Similarly, to train and keep the body trim, we send in the dead organisms or the inactivated organisms inside the system. This provokes the production of antibodies, which in the event of actual infection wards off all those infections.